Our first speaker in this final session, then, is, again, another Caltech scientist here who works in this very subject of consciousness, or at least the study of the brain indirectly there. Dr. John Allman is in the Department of Molecular, Cellular, and Integrative Neuroscience in the Biology Division here at Caltech. He conducts research on the evolution and development of the brain. We have his book, The Evolving Brain, from Scientific American Press there. In which he looks at the reconstruction of the cerebral cortex, the interface between emotion and cognition, the relationship between the anterior cingulate cortex and attention deficit disorder, and depression, obsessive compulsive disorder, phobias, post-traumatic stress disorder. So we've had some overlap. This will be good for today, looking at the brain. And he's the person who's really made the most progress in looking at these spindle-shaped neurons that seem to be related to emotion and social behavior that virtually no other mammals have, exception of a few other of the great apes. So there might be something key to that. John will also be lecturing today on intuition. So from a brain's point of view, from a neuroscience point of view, should be very interesting. With that, Dr. John Allman.
Thank you. First of all, can you all hear me? You can hear me fine. Okay, good. Okay, so I'm actually going to start with a definition, since it's useful to have definitions. And I want to talk about intuition. So this is strictly provisional. It's something that's work in progress. I speak of intuition as a form of cognition in which many variables are rapidly evaluated in parallel and compressed into a single dimension. This compression facilitates fast decision-making. Typically, we are not aware of the logical steps or assumptions underlying this process, although intuition is based on experience-dependent probabilistic models. Instead, we experience the intuitive process viscerally. Intuition operates largely in the social domain, can also be applied to purely physical situations. I have particularly in mind here the wonderful intuitions of someone like Richard Feynman, who thought intuitively about physical systems. Intuition is plastic. It is not instinct, although instinctive feelings may contribute to it. Emotional value judgments contribute to both intuition and to deliberation, which is a slower cognitive mechanism. So I don't want to make a distinction on the basis of one system being emotional and the other not. The emotion contributes to both. The main distinction I want to make is essentially between fast and slow. So we have this peculiar class of neurons, which I think are part of the circuitry for intuition, or for this fast form of intuition, which I think is something which may be a specialization in hominoids. And these peculiar neurons, the von Economo neurons or spindle cells, are located in the anterior cingulate cortex, which wraps around the anterior end of the corpus callosum in this medial view of the brain. Patrick Off and Esther Timchimsky began the modern study of these cells, although they were described in great detail by von Economo back in the 1920s. The other place where they're abundant is in frontal insular cortex, which is this wedge of tissue here that lies between the orbital frontal cortex and the insula. So I suggest that intuition is an important function of this system and if it's made on the medial side. And in contrast, we have dorsolateral prefrontal cortex more preferentially involved in the deliberative thought. Now, if you do a section through the brain, this kind of a section, a frontal section, you can see the location of frontal insular cortex and anterior cingulate cortex in this cross-sectional view. You notice the relative size of frontal insular cortex occupying a large part of the extent of orbital frontal cortex in this plane, contrasted with a very small area of frontal lower cortex in a chimpanzee. Now, the cells that I'm talking about are very large. Note the scale bar here is 50 microns. So this is a von Economo neuron. It can be several hundred microns in extent in terms of its cell body, and then it has an apical dendrite radiating out towards the peel surface and a basal dendrite going down towards the white matter. So this contrast from the classical cells that we see in cortex, which are pyramidal neurons, which are pyramidal in shape, and that shape is imposed by the presence of an array of basal dendrites coming out of their base. And so I think that this population of cells are probably a specialization of pyramidal cells in which all of the growth has been concentrated in a single very large basal dendrite. So the point is that they're located in layer 5, which is a classic output layer of cortex. This implies that they're sending information out, processing out of the structure. They're very large, which is again associated with motor type neurons or with projection neurons. They have many other properties which suggest that they are projection neurons, and we know that they project into the white matter, but we don't know exactly where they go. So they're big neurons that we think are relaying information rapidly out of these two cortical areas in which they exist. They first appear, as far as we can tell from our comparative studies, we looked at about 25 species of primates, and they are not present except in gorillas, humans, bonobos, and common chimps. And from this, we make the inference that their time of origin was about 10 to 12 million years ago, roughly the time of the origin of the antecedents of African apes and humans. So they're a relatively recent specialization. So by comparison, I'll be talking about this in a few minutes, the evolution of bipedality, a characteristic of hominids, is perhaps 6-7 million years old. Now, the monoclonal neurons are a specialization that has arisen in the last 15 million years, and they have proliferated greatly within the human line of descent, and I'll show you that in a moment. Due to their late emergence in phylogeny, the force of natural selection has had only a relatively short time to shape their functioning and integration within neuronal circuits. Consequently, the von Conneville neurons may be particularly vulnerable to dysfunction in a manner analogous to the propensity of humans to suffer lower back, hip, and knee disorders as a consequence of the recent evolution of bipedal posture. So in other words, evolution works just well enough, but it's far from perfect. It has many defects. And some of just as there are defects in the recent evolution of bipedal posture that result in a host of miseries to the human condition and lead people to doctors very frequently, it's also possible that this system, which is both late in evolution and also, I'll show you in a minute, late in ontogeny as well, may be also particularly vulnerable to dysfunction and may conceivably have a role in a variety of neuropsychiatric disorders. I'll talk about one in particular, autism. So we've actually counted these neurons using stereologic sampling techniques in chimpanzees, gorillas, human newborns. This is a very late term, 42-week. This is the normal term infant, and this is a late term infant, a 7-month-old infant, a child of 4 years of age, and a number of adults. And so what you can see is that there are many more neurons in the adult humans than there are in adult gorillas or chimpanzees. So there are vastly more of them. The other thing that you can see is that there is a considerable increment in their number postnatally. Now, actually, because I've had access to the Alcatraz Brain Collection in Washington, which has 1,500 serially sectioned human brains, been able to explore this fairly carefully. But in the von Conneville cells first appear 35 weeks post-conception, rather abruptly. That by birth, there are, say, on the order of 18,000 of them in the sample of 3 that we've looked at thus far. And the number appears to increment rapidly during the first couple of years of life. So they appear to definitely be coming in late relative to other neurons. Most neurons, of course, are formed midway through gestation. And so they're coming in late. We don't know whether they're migrating from postnatal neurogenesis and migrating to their location, or whether they're being transformed from some other cell type. That's something that we're working on, but we don't have an answer to that present. But what we do know is that their emergence as a distinctive morphological type is something that's coming into the first couple of years of life, postnatally. Now, in concert with that is an interesting observation that in the chimpanzees and the gorilla that we've looked at, and also in the adult humans and the 4-year-old child, what we've noticed is that there are 30% more neurons in the right hemisphere than the left hemisphere. And that's quite consistent. So we've seen that in every subject that we've seen beyond infancy. And so what's interesting is what happens in the course of infancy. So in the 38-week full-term baby, they're about the same number in the right and left hemispheres. And then that number increases during the first year of life. And then in the 7-month-old infant, we actually have a larger number, larger ratio, in other words, about a 2-to-1 ratio in the 7-month-old infant. What that implies is that the right hemisphere development races ahead of the left hemisphere, and the left hemisphere then gradually catches up with it. And so what this shows then is that this hemispheric preference, which is so strong in both apes and in older individual humans, that emerges as some sort of developmental process within the first couple of years of life. You're not born with it. And that means that it could be potentially impacted by a variety of events that occur during that period of life. The other thing, of course, is interesting to think about this is that this consistent preference of about 30% more neurons in the right hemisphere is probably associated with other systems that have to do with the experience and interpretation of emotion. Also, it suggests that hemispheric specialization is something that emerged in this system in apes long before the emergence of humans. Now, we know quite a bit about the chemistry of these cells, and I'm going to talk first about the dopamine receptor, which is very strongly expressed on these cells. This is the dopamine D3 receptor. It's one of 5 dopamine receptors. And this particular one, D3, is the high-affinity dopamine receptor. They're from D1 through D5. The D1 and the D5 are low-affinity receptors. So this is a receptor that has 100 to 1,000 times higher affinity for dopamine than the others. And it's very strongly expressed on the von Economo neurons. And this is a slide that Terry showed a few minutes ago. And the point I'm not going to go through all that, but just simply say that note this slide here. In a situation in which you present a stimulus that is known to be associated with a reward, then you get a volley of activity in the source populations for the dopamine input, which is the ventral tegmental area. So this is the activity of the cells which are projecting into this system, the source of the dopamine. Now, Wolfram Schultz did a really wonderful experiment. It's an extension of this, in which he signaled by different symbols the probability of reward, so that they range from probability of 0, probability of 0.5, up to a probability of 1. And so when you present a signal that indicates a probability of reward, you get, of course, an initial volley of activities, which is essentially what was shown in the previous figure. But in about a third of the neurons, you also see this ramping up of activity that precedes the receipt or non-receipt of the reward. This is associated with uncertainty. So this second signal that's being communicated by these neurons is one of this anticipation. The excitement mounts as you're waiting for whether or not you're going to receive the reward. And I think that this is a different kind of information. And because it is a relatively smaller signal, my conjecture is that is picked up by the higher affinity receptor. So this would be picked up by the low affinity receptor. And then this more subtle effect would be picked up by the high affinity receptor system. So it's interesting that Hugh Critchley, working completely independently, did an analogous experiment in humans in which he had a gambling task, in which the subjects are gambling for cash at a card game. And he varied the degree of uncertainty in that task, and he measured the activity in an fMRI study of anterior cingulate cortex and frontal insular cortex. Now, he didn't know anything about the von Economo cells when he did this. It's remarkable that it highlights the same structures. And the point being is that as you increase uncertainty in this task, you increase the level of activity during this period of expectation. It's the same phenomenon, in other words, that excitement mounts as you wait for whether you're going to get a reward or not. So uncertainty is a powerful determinant of the activity of this system. Now, John O'Doherty has done a very elegant series of experiments in which he did also a gambling task. And in this gambling task, you could make a wager, and you would either get a cash reward or penalty according to the outcome of that wager. And then you could potentially change your wager to a different form of wager, a different kind of bet. And so if you just simply make your wager and receive a reward, that seems to be actually associated, the reward signal actually reduces activity. If you make a wager and receive a punishment, receive a penalty, that's also associated with a reduction in activity in both frontal insular cortex and anterior cingulate cortex, same places where the monocondymal cells live. If you make a wager, take a penalty for that wager, and then decide to switch to a different wager, that is associated with an increase in activity here. So it's like the adaptive decision-making process. Taking a hit, deciding to try something else. That's associated with specific activity of FI and ACC. Rebecca Elliott noticed in similar types of gambling experiments that area FI is active when the subjects report they have a sense that their luck is about to change. So there's a powerful association with reward and particularly the uncertain component of reward. Also, we have another neurotransmitter receptor which is strongly expressed on these cells, which is the serotonin 2B receptor. And the serotonin 2B receptor is very interesting because it is very rarely seen in the central nervous system. Instead, where it is seen is in the mesenteric nervous system, where it regulates gastric motility. And so it literally is visceral feeling. It's what's generating the differences in visceral feeling. And so this system, the strong expression of the serotonin 2B receptor on these cells strongly implicates these neurons in a relationship with the gut. And so the other thing that's interesting about this is Peter Dian, who Terry mentioned before, has a very interesting theory about serotonin that suggests that serotonin is dopamine's evil twin. That serotonin is the signal for expectation of punishment, potentially. And that links then to this function in the peripheral nervous system, in the mesenteric nervous system, which has to do with your gut churning, literally, which is a commonly used indication that you may get punished soon. Anyway, so that's a second system. So I'm suggesting that there's maybe something like a push-pull relationship between expectation of reward and expectation of punishment in this system, an integration of those two probabilities. Now, there is a third neurotransmitter receptor that's also strongly expressed on these cells, which is the vasopressin 1A. And from a beautiful series of experiments initially started by Sue Carter and then pursued by Tom Insel and Larry Young, have shown that vasopressin, and particularly the vasopressin 1A receptor, are necessary and sufficient for mediating the formation of social bonds. A beautiful series of experiments in rodents. So it's interesting that we find this also on these cells. And that suggests is that there's also a social affinity component to this process that's impinging on these cells. Now, we have some other interesting evidence for that. The one thing, I'll come to that in a second, but one point is that social situations which involve uncertainty, such as embarrassment and guilt and lying, all activate frontal insular cortex, rather specifically. Now, with respect then to the social bonds, we have this remarkable experiment by Bartels and Zeki, in which they did several different experiments in which subjects looked at loved ones, both lovers and also women looking at their children. Both activate FI and ACC in the spindle cell containing areas. And FI, but not ACC, is also activated by the sounds of infants crying. So anyway, we have a whole variety of different things, and there are a great many more that I could mention, that implicate this structure in sort of the intuitive components of social interactions, and also in uncertainty. And in contrast to this other system that I think is involved in deliberation. Now, I'd like to suggest that autism, or the whole class of disorders are called the autistic spectrum, may represent what it might be like to have this system not work so well. And so this comes from, when we interact with another person, we usually create a mental model of how that person thinks and feels. This model is a synthesis of our fast intuitions and our slower deliberations based on fact, logic, rules, etc. Describing subjects with Asperger's syndrome, Clint and Volkmar observed their deficient intuition and lack of spontaneous adaptation are accompanied by marked reliance on formalistic rules of behavior and rigid social conventions. Our hypothesis is that the Bonacama neurons are an important part of the circuitry responsible for intuition, and that these neurons are dysfunctional in autism spectrum disorders, resulting in defective intuition. Or putting it a different way, they have to rely much more heavily on the deliberative process. And this is sort of revealed, I think, in a wonderful statement by a very high-functioning autistic individual, Campbell Grandin, who said, "Each day I collect more data to place in my library of experience. When I encounter a new social situation, I have to search my memory for a similar experience that I can use as a model for my next action. I have a very difficult time when I'm confronted with unexpected social surprises. For common social interactions with clients, I use pre-programmed, pre-rehearsed logics, pre-rehearsed responses. Everything is done with logic. So this is the deliberative system. And a high-functioning individual, that's what they have to work with. So Clint and Volkmar point out individuals with Asperger's, which is a form of autism, typically cannot avail themselves of their formal social knowledge in a quick pace, simultaneously shifting social situations. They often miss the tempo of interactions and lose any possibility of rapidly adjusting themselves to the forever shifting social and community demands of others. In other words, speed is important and rapid adjustment to changing contingencies. This is notably true for social situations, but it's not restricted necessarily to social situations. So Asperger himself actually commented on another thing, which is related to this. Another characteristic of autistic children is the absence of a sense of humor. Autistic children are rarely relaxed and carefree and never achieve that peculiar wisdom of deep, intuitive human understanding that underlies genuine humor. So I was inspired by this to actually look at humor. And so we took 50 cartoons from the Far Side of New Yorker, and we dehumorized them by deleting little things such as, yes, we're open to the vulture side here. And so then we put subjects in the scanners. So we had 100 cartoons, half of which are dehumorized. And then we asked the subjects while in the scanner to rate how funny they thought the cartoons were. And specifically, area FI is activated in proportion to how funny they think their humor is. So that is pretty clear linkage. Now, there are a number of other linkages. I just alluded to humor. I also like to say one more thing about humor, and that is that Freud actually suggested that humor is a way of relieving tension. And I think that may be an essential for the functioning of the intuitive system, to periodically recalibrate it, or else you would build up a series of things that would be malfunctioning. Anyway, so humor activates both FI and ACC. It's certainly abnormal in autism. Social bonding activates FI and ACC. It's decreased or abnormal in autism. Empathy activates ACC and FI from studies by Tanya Singer and her colleagues. That is said to be decreased or certainly abnormal in autism. Trust also from a study by Tanya Singer, activates FI, but apparently not ACC. And curiously, Ralph Adolph showed that trust is increased because of difficulty reading social cues in autistic subjects. Finally, Barron and Cohen showed that discrimination of mental states of others activates both FI and ACC, and it's decreased in autism. So and there are quite a few more I could add to this chart, but it's I think, pretty massive evidence. Now, then I'm just showing this just to remind you of where FI is located, a normal subject. And And what we've done now is we've looked at two autistic children, an autistic boy and an autistic girl who are 9 years of age each. And we've looked at where the von Economo cells are. We saw something that was rather surprising to me. Now note this is where it is normally. And this shows the plotting of actually each individual cell. And it's located more medially than normal in these individuals. And it's also the cells are normally located in layer 5. So what you would see in a normal individual is a band of cells here in the gray matter. But instead what you see them is heavily in the white matter, and you see rather large concentrations of them. You see more of them in the autistic subjects, but they're in the wrong place. And that suggests to me that there's some sort of a defect, whatever it is, in their migration or in their differentiation that occurs in the first few years of life, that is not occurring normally in these individuals. And that for whatever reason, they're ending up in these clumps, so that this actually, when you look at it in a microscope, it looks like a log jam of von Economo cells that have all sort of backed up behind some invisible barrier. Anyway, so von Economo neurons may relay a fast, intuitive assessment of complex social situations to facilitate the rapid adjustment of behavior in quickly changing social situations. Thus, they may be a neuronal specialization linked to the increased complexity of hominoid and especially human social interactions. We hypothesize that these specializations enable us to reduce complex social and cultural dimensions of decision-making into a single dimension that facilitates the rapid execution of decisions. Other animals are not encumbered by such elaborate social and cultural contingencies to the decision-making and thus do not require such a system for rapid intuitive choice, at least at such a level of complexity. Because of their late emergence in phylogeny and ontogeny, the von Economo neurons may be particularly vulnerable to dysfunction. Now finally, why would natural selection favor the evolution of neural circuitry responsible for embarrassment, guilt, empathy, and cooperation? The rational choice economists would sort of deny that this is even important. So Robert Frank, who is himself an economist, wrote a wonderful book called Passions Within Reason, the Strategic Role for the Emotions. And he points out there, the moral sentiments, the things I just talked about, may be viewed as a crude attempt to fine-tune the reward mechanism, i.e. dopamine, to make it more sensitive to distant rewards and penalties. In other words, by distant, he means distant in time. So that situations in which an individual would forego a reward in the interest of long-term maintenance of a relationship or would accept a penalty in the interest of maintaining a relationship. But the important issue is distant in time, not immediate reward, foregoing that immediate thing. I think it's a really central insight. And so apes and humans are long-lived and their offspring mature slowly. Therefore, the future is important. The prospects for future success depend on mutually beneficial associations, reciprocal altruism, and this network of cooperative relationships constitutes an individual's social capital. And I would maintain that that's the foundation of financial capital as well. And to establish an effective social network, one must have some way to determine who will be a faithful partner in future interactions. That's the crucial problem that you have to figure out. And that's done both through deliberation and through intuition. Such determinations arise in part from one's intuitive assessment of the likelihood of potential partners will be empathic to your misfortunes, will be deterred from defaulting on obligations to you by feelings of guilt. Hard to do. Maybe we do just a little bit better than chance, but well enough. So that's, I'm going to leave you under that. Yeah.
Great. Thanks. Good job.